My research here is studying a disease called leishmaniasis, which is caused by a protozoan parasite. It's transmitted by sandflies and found mostly in developing countries, South America, lots of Africa, the Middle East, but it's also in recent years spread to places like Australia and South Italy as the climate changes and mosquito and sandflies are able to travel further. So the disease itself can manifest itself in several ways. I work on the cutaneous form, which is where the parasite is confined to the skin, and it infects dermal macrophages and then can cause ulceration and open lesions, which can leave people open to other infections, but also if they do get the treatment and it heals, it can leave them with massive scars and cause stigmatization in some of the cultures that they live in. So the current models for leishmaniasis are generally thought of to be the in vivo model or an in vitro model, which is macrophages on the bottom of a whale plate, usually stuck down in 2D, and then that's treated with drugs. So the models themselves lack complexity. So the in vivo model is obviously in the mouse, and there's many papers about the difference between mouse and human biology. And then when it comes to the in vitro model, the biologically relevance of macrophages stuck down in 2D, so they don't form the right conformations that you'd find in the body. They've got no stimulation of other cell types or of the interstitial fluid flow. And as a project, we're trying to increase complexity in our model by firstly introducing the interstitial fluid flow. So the cells are under the same shear stress, oxygen tension, and supplement delivery that you would find more closely in a biological situation. We're hoping to achieve the fluid flow caused through the system to match interstitial fluid flow, and hopefully this will provide a more biologically relevant environment for our cells, so that the media flow across the top of the surface of the cells will be at the same speed as they would feel in the body, generating the same shear stress and biological cues that you would gather from such movement. What the Quasi Vivo plates do for us that static models don't is allow us to introduce the fluid flow and vary the rates so that we can measure different responses to different fluid flows. Importantly for us, we can measure interstitial fluid flow rates across the bottom of the whale and replicate what the cells would feel more in vivo. My favorite thing about the Quasi Vivo system is the design of the plates. So they're so easier to work with when they're all connected together in one place. Easy to move back and forth, generally easy to transport and use as a whole. So it's much nicer that they're in a complete package and you don't have to worry about all sorts of attachments and nothing like that. Another thing that I really like about it is the adaptability of the assay design. So I can connect this plate in 2 sets of 3 or as a whole 6, and this allows me to produce more data if I need or scale back the experiment. I can introduce multiple variables within the same plate, and just being able to connect it in different ways allows for me to design my experiment around it in a much easier way than I'd previously have to think about designing my experiment for the plate instead of changing the plate to fit my experiment.